Oh, is it not showing up in the class at all? That is odd. I thought I had all those in there. Dice kinetics, equilibrium practice problems. Oh, it's not in there. I am sorry. I thought I had that in there. I guess I have all the other ones, but not that one. I will get that put in as soon as we are done here. I have the files pulled up here where everyone will be able to see them as we're going through, but I'll get that put up as soon as we're done. Okay. Yeah, so yeah, I thought I had it in there, and it's just not there. So let me start this back up again. Okay. So this is the Word file. There's also a PDF that's just the same thing, just not Word. And then there's also going to be what I know is everyone's favorite thing, an Excel file. Everyone loves to use Excel. And yes, that is sarcasm. But we need this Excel file to get data. So this particular lab, like I mentioned a minute ago, is kinetics again, but it's still it's a good thing to work on, and since we've been doing kinetics for a while now, hopefully it'll be a little more straightforward than maybe it would have been if we had tried this, the first week that we were doing kinetics. This one is going to be a little bit different, however, in that while we are doing kinetics, we're not going to be making big graphs with a natural log of something versus time and 1 over something versus time. We are going to be determining the rate law using multiple trials that we're going to get from the Excel sheet. So it's the same topic, but not quite the same. It's not quite as data intense, I guess you could say. It's not quite as intense in terms of making graphs and making sure you have the right equation and pulling things out of the equation and doing math to them. It's not going to be quite the same as that. So let's just go ahead and, since I didn't make it available to you before, we'll go ahead and we'll just read through this together and talk about it a bit. So the iodine clock reaction is a famous experiment first performed by Hans Landolt. I have no idea if I'm pronouncing that correctly, back in the 1800s. In this lab, we will study the kinetics of this reaction, which is between iodide and persulfate. So iodide is the anion form of iodine. I'm going to try to be very clear in my pronunciation because it is very easy to get those mixed up, and we will have both of them in this reaction. So the iodide reacts with persulfate, and when it does that, it makes plain old sulfate, which we're very familiar with, and iodine. So it turns the iodide back into just elemental iodine. This reaction, iodide is oxidized to elemental iodine by the reaction with persulfate. You will be able to time this reaction by using 2 additional reagents, thiosulfate and starch. And it makes a little point here of saying that the thiosulfate is not the same as persulfate. So just be careful as you're going through and you're doing your work that you don't get those 2 mixed up. So the persulfate is the 8 oxygens and the thiosulfate is 3 oxygens. So with those 2 extra reactants, you have these 2 essentially side reactions, if you want to call them that. So when you have this initial reaction taking place, you have iodide reacting with that persulfate, and it makes iodine. If you just had the starch and you didn't have the thiosulfate, what would happen is the moment that the reaction starts, which is just, the second that you put the stuff, you mix them together, you would get a deep, dark blue color. It's because starch, when it's in contact with iodine, it essentially wraps itself around the iodine, almost like a chain or a snake or something. It wraps around the iodine, and that particular complex interacts with visible light to make a blue color. This is a very common way to test for starches, actually, is to mix it with some iodine. And I guess conversely, if you wanted to test for iodine, you could mix it with some starch. But usually it's the other way around. And so as soon as you start the reaction, it would turn that dark blue just immediately. To keep that from happening, what we do is we add in that thiosulfate as well. And the thiosulfate essentially blocks that reaction from taking place. It gets in there first. The iodine prefers to react with the thiosulfate instead of the starch. And so as long as there is thiosulfate around, the iodine reacts with it first to form the iodide back again, and this, what's that called? Does it even give the name in here? I don't remember what that ion is called. But it forms this S4062- complex, which is not dark blue in color. So we have this system that as long as there is thiocyanate still around, it will not turn dark blue. But as soon as the thiosulfate is used up, we will get a bright blue color, or this really dark blue color, I should say. And so we're going to use this as a way to track how quickly this reaction happens. Because this reaction is what's making the iodine in the first place. And so this reaction is essentially using up the thiosulfate. And so we'll be watching and seeing how long it takes, the actual time period that it takes for the thiocyanate to get used up. And if it gets used up really fast, so we see that blue complex really quickly, that means this reaction was very fast. If it takes longer, then that means the reaction was going slower. So the kinetics are slower. And so that's the general idea behind this reaction. And it's a pretty standard reaction. I've done it multiple times in the lab. It's been a while since I've done this online version of it. So I think all that stuff that I just talked through is pretty much just summarized here, I believe. I don't think I want to read through all that. Initial concentrations, I saw the same. Well, there's a little bit extra there, so I guess we'll go ahead and read through this. So up to here maybe. So it's talking about pretty much what I just said up to here, and then it says, thus, by addition of a small amount of thiosulfate, color will not be produced until the thiosulfate in the solution is depleted, after which the deep blue or black color will appear. If the initial concentration of the thiosulfate in each trial is the same, then this provides a useful method to time the reaction, because we will know exactly what concentration of iodine was produced before the color changed. So essentially it's just saying if you have the same amount of thiocyanate each time, you can vary the amounts of the other stuff. You can vary the amount of iodide, vary the amount of the persulfate, and any changes in the time that it takes for the reaction to occur are not due to the thiosulfate, because that's the same every time. It would be due to whatever change you made to these two. So it goes through an example here. It says, for example, suppose you use a constant amount of 5 mils of the 0.0055 molar thiosulfate and a constant total volume of 65 mils. The trial took 23.1 seconds for the mixing until the color change, so the diluted concentration and rate would be calculated like this. So the dilution is because while we used 5 mils of this concentration, we mixed it in with other stuff. We mixed it in with some iodide, We mixed in with some thiosulfate, or sorry, persulfate, and so it didn't stay as 5 mils in this concentration. It got diluted down to this concentration because we diluted it to 65 mils when it was 5 mils before. And so then if it took 23.1 seconds for the blue color to show up, that tells us then that this concentration got consumed in this many seconds. And if you remember, rates are always just a molar change and then the time that it takes to get that molar change, so it's a molar per second. And so it is just literally the molarity divided by the time. And so that would be the rate. And so you're going to be doing lots of these calculations when you're working through this lab, both finding diluted concentrations and finding the rate from the data that's given to you. So, in this lab here, the things that you're going to be working with, the simulated stuff that you can mess around with, is the persulfate, which is ammonia persulfate. We don't really care about the ammonia though, it's just there as a counter ion, it doesn't really do anything. We have Ki, potassium iodide, that is our source for the iodide, and just like the ammonium, we don't care about the potassium, it's just there as a counter ion. It's going to disappear essentially. The thiosulfate source, and then the starch indicator. And truthfully, we don't really do anything with the starch indicator. It's just automatically included, so to speak. It's not, I mean, it's just a calculation. So it's automatically already in there, and everything's good to go with that. So, what we're trying to find, this is our objectives here. We are trying to determine the reaction order with respect to iodide. We are trying to determine the reaction order with respect to persulfate. We are trying to write the rate law and the overall reaction order. And then we're trying to determine the rate constant at room temperature. So those are what we're trying to find. Now, this particular experiment is what's called an inquiry-based experiment. We do 1 or 2 of these in my Gen Chem 1 class. I don't think we've done any of these in this class yet. Essentially what an inquiry-based experiment is I tell you what you have to work with, I tell you what I want you to find, and I give you as little information as possible in terms of instructions as how to find it. And it's your goal to work out, given what I have and what I'm looking for, how do I make the connection in the middle. So I do give you some hints here, and I'll talk through some stuff as well as we look through the rest of the lab file here. But my goal here with these experiments, these inquiry-based experiments, is I don't want you to have a do this and then do this and then do this and then do this type experiment setup. Those are useful sometimes, they're good to have sometimes, but it's also good to have the ability to develop your own procedures, to work out yourself. Okay, I know that I need this information, how can I get that information? So let's see here, we have some helpful hints. It says use the same amount of thiosulfate in each trial and the same few drops of starch indicator, and it says the indicator is added automatically in the simulation. Does it actually give you a spot to put that in? I don't think it does. Yeah, so you don't even have to worry about the starch indicator. There's not even a spot for you to tell it how many drops to put in. It's just automatically in there, and it's automatically the same each time. If you were doing this in lab, you would definitely want to use the same amount each time. So use the exact same amount of thiosulfate each time. Is it technically possible to do it with different amounts? Yeah, but you're just going to make the math egregious. It's going to be a lot easier if you just keep it consistent. Remember that you should only change one variable at a time. You must have multiple trials, at least 3 each, where one reagent is changed while the other is kept the same at the same concentration. When we've done things like this in the lecture videos, we only used 2 experiments. So we would have 2 tests where one thing was kept constant and the other one changed. In this one, I want you to have 3. So you would have 3 experiments where something is kept constant and then the other thing changes, and then you would essentially flip that. And so then in the next 3, you would have the thing that was changing before be constant, and the thing that was constant before be changing, just to get some better data, so we can get an average. Getting an average of 2 values is not terribly useful sometimes. So the next one there, it says maintain a consistent total volume, somewhere between 50 and 60 mils for your trials. This will make your calculations easier. Add water if you add less volume of any reagent, such that the total volume is consistent. Now notice here, I'm not telling you you have to have 50 mils or you have to have 60 mils or whatever. It's up to you, whatever you want the constant volume to be, but you do want to keep a constant volume. And again, do you have to? No, you don't have to. You could do this and have a different volume each time. You're just going to make the math a lot harder on yourself. It's easier if you have the same volume, so when you're going through and doing these dilution calculations, if you use 60, for example, each time, then just this bottom one is always 60, because that's the total volume. If you're unfamiliar with where this equation came from, by the way, this is just a slightly rearranged version of the, is it going to let me write on that? Maybe it's not. It's a slightly modified version of the C1V1. Nope, it's not going to let me write on it. C1V1 is equal to C2V2, or M1V1, if you learned it that way. It's equal to M2V2. It's the dilution equation. So if you're unfamiliar with the dilution equation, you can go back and watch my Gen Chem 1 videos over that. So this is just a slightly rearranged version of that. Okay. The next tip here, the next hint, says the concentrations used in all the rate law calculations are not simply the molarities listed on the stock bottles. Each reagent is diluted when mixed into the reaction mixture. You must take this into account. So again, lots of dilution math is going to be done here. Because over here in this table, you're going to be putting in a number of milliliters, and then it's going to give you the time that it takes. However, when you go through and you make your second table, which we'll talk about in just a minute, you're going to want to put in molarities there, not mils. You need molarities in the second table, not milliliters. Okay. So just to warn you beforehand, the reason why we said you want the same volume each time is because each one of these squares is going to be a dilution equation that you're going to have to work out. Well, I guess not the water. We don't really care what the concentration of the water is. But for these 3, you're going to be working out what those concentrations are. The thiosulfate actually will be the same each time, because again, I suggested that you make it the same each time. And if you remember, I said technically you don't have to make it the same each time, but you're just going to make the math harder on yourself. That's what I was referring to. If you wanted to have it different, you could. You just have to do a whole extra dilution equation for each one. If it's the same each time, you just have to do it once and then fill them all in. Okay, so the last one here says, and by the way, this simulation will not necessarily find the same conclusion as the real experiment. We've changed things up. So if you get to the end of this and you're like, I don't know if I did this right, and then you Google the experiment and it tells you something completely different than what you found, that does not mean that what you found on Google is what I'm looking for. you should trust your data. You should use what you got and put that in there, because what you have is not necessarily the same as what you would actually get in a lab experiment. I'm looking for the process here, not for the final answer. Okay, I know we've been going for a little while, but this is something that we need to talk about, just because I know we've had some struggles with labs the last couple weeks, so I want to make sure that I'm doing the best possible to explain what's going on here. Are there any questions so far before we get into the last section here? Any questions? Okay, if you're typing one in, just keep typing it. I'll pull something else up here real quick. Maybe I am. Why is that not opening? There we go. That was weird. Okay. So let's look at that last section here, the data analysis. So it says, converting your data. The simulation provides your raw data in units of mils of each reactant, not molarity, and seconds, not rate. Therefore, the first task is to construct a new data table on your own, either just on paper or in a Word document or an Excel workbook. Any of those are fine. Like I said, we're not going to be doing big graphs or anything, so if you want to do it in Excel, that's perfectly fine. If you would rather just do it by hand, that's perfectly fine too. Either one is fine. So convert these values into the proper units. If you wish to copy and paste the data into a new document or worksheet, make sure you use the paste values or paste text only options. If you just normal Control-C, Control-V, it's going to try and copy all the equations and everything that are already in the Excel sheet, and we don't want that. We just want the numbers. So it says, construct a new data table with the columns trial, concentration of persulfate, concentration of iodide, rate, and rate constant. To convert mils to meters, use the dilution equation. To find the rate, follow the example on the first page. So it's talking about these here. And so, like I said, this is actually just the dilution equation there as well, just slightly, it's this with both sides divided by the V2. So you're left with just M2 over here. So this 4.23 times 10 to the negative 4 is M2. This is M1, V1, and V2. So it's just the dilution equation. So you're going to take your numbers from this, convert them into the proper units, molarity, and find the rate using this equation as well. So it would be the concentration there divided by the time that it takes, and that time will be given to you in the Excel sheet, which we'll do an example in just a second. Okay, it says leave the rate constant column empty for now. You won't be able to determine the values until later. So then we have determining the reaction order for each reactant. To determine the reaction order, we compare pairs of trials where only the species in question has changed. So it says you may have derived this lovely equation in lecture. We did not derive that in lecture. We just looked at examples that were simple enough we didn't need an equation. But the way this equation works is, so if you remember back, oops, not that one, this one. So back here, we had tables like this, where we had some things that didn't change and then things that changed. And so we said, okay, this doubled, and we saw the rate double, and so therefore it was first order in respect to ammonium. Now, because the numbers here were so simple, it was easy to see that that's how it worked out. Our numbers are not going to be that way. our numbers are not going to be 0.01, 0.02, 0.04. Like, they're going to be more complicated than that. And so to do what we did here just in our heads, just by looking at it and saying, oh, doubled and doubled, we need an equation for that, and that's what this equation is. So the order with respect to whatever it is that we're looking at, whatever reactant it is, that's the m there, that is going to be equal to the log of the rate for the first trial divided by the rate of the second trial, and that divided by the log of the concentration for that first trial divided by the concentration for that second trial. So in our example here, to find the order with respect to the ammonia, we would take the rate for trial 1, since the ammonia is the one that's changing here. So it would be 5.4 times 10 to the negative 7, that would be R1. 10.8 times 10 to the negative 7 would be R2. Concentration 1 here would be 0.01. Concentration 2 would be 0.02. So if you plug those numbers in, you should end up with 1 here for the m value. And so that's just the exact same thing that we're going to do here in this lab, except our numbers, like I said, won't be quite so neat. But if we just plug them in, we don't care that they're not quite so neat. Okay, so continuing on here, it says you should have multiple pairs to compare. For example, if trials 1 through 3 all use different concentrations of A, but the same value for B, then you can do this equation for trials 1 versus 2, 1 versus 3, and 2 versus 3. The true value for a species should be the average of all comparisons. Report the average reaction order for each species round to the nearest whole number. And they should be pretty close. Like, you shouldn't be getting like 1 and 2 and 1.5. Like, they should be pretty close, but they might vary a little bit. So, like I said, you should have 3 for each of them. 3 comparisons to make there. Writing the rate law. The rate law always has the generic form. The rate is equal to k times the concentration raised to some coefficient. Now that you know the reaction orders for both reactants, excuse me, writing the rate law is as simple as inputting those values into the equation, leaving everything else as variables. So report the rate law for this reaction, showing the values for the reaction orders for both species. So you would have just the rate is equal to K times iodide raised to whatever number, probably 1 or 2, but I won't, I actually don't remember what this is set to give you. And then per sulfate raised to some number. Now that you have written the rate law, rearrange the equation to solve for the rate constant. Use this to solve for the value of K for each of your trials, then compute the average value. Report the average value for the rate constant K with proper units. So essentially you're going to do the same thing that we did. Let's see, did we do that in this one? In one of these examples, yeah, it was this one. So in this example here, we solved for K. So we took data just like you're going to have for this lab, and we found the order with respect to each of these materials, each of these reactants, and then we solved for K. So you're going to do the exact same thing in this lab that we did here in this practice problem. Only in this practice problem, we only did it for one trial, and in the lab, you're going to do it for all of them because they will all be slightly different. And so you're going to get all of those and then take an average. So if you have 6 trials, which you should, or yeah, at least 6 trials. There's spots for 8 here, but I think you only actually have to do 6 because it only asks you to have 3 constant for each of the 2 reactants. So I'm not entirely sure why it gives you spots for 8. Anyway, so you would have 6 different constant values. So just add all those together, divide by 6, and there's your average. All right, so then, in conclusion, the data table from the simulator shows your trials and raw data. You converted the data table in units of molarity and rate, or into those units. Your calculation to determine the reaction order for each reactant, the rate law, the calculations in determining the rate constant for each trial, and the average rate constant. This is what I'm going to be looking for in what you turn in to me. So if you're missing any of these things, you're going to lose points, for sure. If you have all of those things, and everything looks fine, there's no mistakes, then you'll get full credit. But there can be mistakes made, but at the very least you should have all of these present somewhere in the lab. Okay, so just real briefly here, just to show you how this Excel sheet should work. Okay, let's see. What were the suggested volumes? I forget. I know it said 3 to 5 for the thiosulfate. I'll just go ahead and do 5 for that. Let's see, what were the other suggested volumes? Or did it even say? It might not have even said. Yeah, it doesn't even say, which is perfectly fine. So we'll just pick some ones. We'll just pick some random numbers. Because like I said, it's a inquiry experiment, so you don't get lots of instructions. So just, this should be, since this is 5, if you remember I said the thiosulfate should be the same for all of them. So just make all those 5. We'll just pick a random value for the persulfate. Let's say 15 for that, and we'll do 25 for this. And we want to have somewhere between 50 and 60, if I remember correctly, is what it suggested for the total volume. I only have 45. I'll go ahead and I'll do 55 for each of my experiments. And so I have my values input there. And again, it doesn't actually matter what each of these things are. I'm just going to pick numbers that I want. And as long as I have the same total volume at the end, that's all good. So then I go here and I put 1, and it tells me how long it took. So it was 44.23 seconds. That's how long it took for the reaction to happen. Okay, so at this point, I'm just going to do the same thing for the rest. Remember, I need to have 3 that are the same for each of the reactants, so I would make sure that I have 2. I'm going to go ahead and make the iodine constant for the first 3 here. So then these I can just make whatever I want, as long as the total volume can still be 55. So I can't do like 100, but any volume that would work. So I could do 18 and I could do 21 if I wanted to. That's perfectly fine, as long as I then add water to make sure that I get back to 55 mils total. And there's how long it took. And so then I would just do the same thing again here, but now I would make sure that the persulfate stays the same for each of them. So maybe I'll have 10 for those 3 for the persulfate, and then I would have some different volumes for those. And again, as long as I then make sure that the total volume is 55. Let's see, 29 yet. And so that's how you get your data. Now, obviously, nobody can use this data since I just made it up. You get to make up your own numbers here. You don't have to have every single number different, but you can't have the exact same data that I have here. So you'll get your own data, and so then this will be what you will transfer over, and you will essentially just be making your own version of a table like this, like we did in those practice problems. And you'll use this data to do that. So again, these two columns are what you will be converting into molarities using the equation from page 1. This column is what you'll be using to find the rate, given the time here and the concentration of the thing that you're changing. That's how you'll find the rate. And so that is how you will fill out these things here, concentrations and rates. Phew, okay. We lost a few people in the process. Are there questions about the lab? No questions so far. Absolutely, yes. You can email, and like I said, I did record this, so I will. I'll probably just go ahead and upload it. Normally, I wait until someone asks for it to upload it, but I'll probably just go ahead and upload it either way. Just because it's not terribly time-consuming to put it up on YouTube, but it is one extra thing to do. But I'll go ahead and do that. Any other questions? I'm glad that it was helpful, Naomi. Doing yeah, I am not your typical YouTuber. I will not say like and subscribe, share with all your friends. I mean, I guess you can if you want to. I don't remember actually what the context was for that, but I do remember doing that. I tried to have a little bit of fun when I was making the videos. One of these days I'm going to make another video, and I am going to like do some kind of typical YouTuber, like and subscribe, if you enjoyed this content, and go to my Patreon or something like that. I don't have a Patreon, by the way. But, just for kicks and giggles, we'll do that at some point. Yeah, pretty much all of them are. There are a few videos that are less than 10 minutes. But no, I am not trying to optimize my videos for the YouTube algorithm. I have had a few just random people like show up at my videos and make comments or something like that. Have a decent bit of spam as well. Where someone will just put in some random comment with a timestamp and then some gibberish. I'm like, I don't know what the point of this is, but I'm deleting it. Anything else? No? Okay. Go ahead and stop the recording then.